So welcome back, students. For Chapter 2, we'll be talking about the chemical composition of the body and how these compositions relate to physiology. And this is really kind of like a chemistry / biochemistry portion of the course where we'll go through different molecules like proteins and carbs and lipids and so on. So first off, these are all the topics that we'll be covering. We'll talk first about atoms, and then molecules, and then solutions. So which one of these molecules can dissolve in water to make a solution and which ones do not. And then we'll talk about the organic molecules, which is related again to biochemistry, which are the molecules that we find in our bodies. So we'll talk about carbs, lipids, proteins, and nucleic acids. First off, we have to kind of go way back to high school to remember what are atoms, neutrons, and protons, and so on. So the atom is the unit of matter that forms all of the chemical substances. It is basically the smallest building block of anything of matter. Okay, that matter could be a gas, liquid, or a solid. And if you break it down into its components, the atom is the smallest particle that would have the characteristics of that molecule. If you break down the atom, you'll get the subatomic particles, which are basically your protons, neutrons, and electrons. And these do not have any properties of your atom, no longer has properties. So I'll give you an example. So for example, hydrogen molecule H2 is made out of 2 atoms of hydrogen. But if you break down one of these hydrogen atoms, you are going to find protons, neutrons, and electrons that are the exact same particles that you will find in an oxygen atom, for example. So the protons, neutrons, and electrons of hydrogen no longer carry the same characteristics of hydrogen. Now, there are 2 different things that you want to keep in mind. First off, that the nucleus, which is right in the middle of the atom, has the protons and neutrons. Protons are positively charged. Neutrons are neutral, meaning they have no charge. Orbiting around the nucleus are electrons, and those are negatively charged. Okay, so here you can see that image where in the nucleus you have these positive protons, those are in red, and these white neutral neutrons, and that will make your nucleus. Orbiting around them in these orbit shells or in the electron shells are known as your electrons, so these are negatively charged. In your first level shell, you can get up to a maximum of 2 electrons. And then if you want to add more electrons, you'd have to now go to the next electron shell and then start adding more to those. And the maximal amount that you can have in the second level are 8. And in the third level would be 8 and so on. And you'd have to add them in that order. So you have to add, 2 here. And then if you have 3 more, you go on and add it to the second shell and so on. So it's a very organized way of adding electrons. And in order for an atom to be constant or to be stable, the outermost shell would need to have the maximal number of electrons. So if the maximal number here on that second orbit is 8, then that in order for this atom, which is a nitrogen atom, by the way, in order for it to be stable, you want to have 8 on here. But you see here we have 1, 2, 3, 4, 5. So in order to become stable, it would have to get 3 extra electrons. Okay, or get rid of all 5, but it's easier to gain the 3 electrons as opposed to losing the 5 electrons. Okay, so we talked about the nucleus having the positive protons and the neutrons. That is really the weight of the atom. That is known as the atomic weight. So your atomic weight is the weight of the nucleus, and we don't really take into consideration the weight of these electrons. Okay, the atomic number, though, on the other hand, is the number of protons. Okay, so in order for the atomic number, which is kind of like the social security number or the thumbprint of an atom, that never changes Okay, so these number of protons are always going to be constant, and by definition, the atomic number would be the number of protons, while the atomic weight is actually the weight of the whole nucleus. So that includes protons plus neutrons. Okay, in most cases, atoms have an equal number of protons and an equal number of electrons. So why don't we just say that the atomic weight, by definition, is the number of protons or the number of electrons? Well, that is because the number of electrons actually can vary. So remember we said in order for this atom to stabilize, it could either gain 3 electrons or lose all 5. So the number of electrons can change. Okay, but since the atomic number, which again is the social security number of this atom, has to be constant no matter how many electrons it has, so by definition, your atomic number is going to be the number of protons, because no matter what happens to these electrons, the number of protons will always be constant. Okay, so going on to the atomic number again, this is very unique to every atom. Okay, and again, by definition, it is the number of protons. And although the number of protons tend to be equal to the number of electrons, but since the number of electrons can change, is not constant, we are going to define the atomic number as the number of protons. So a hydrogen atom has only one single proton in the nucleus, so the atomic number of hydrogen is 1. And you know, so on for the rest. If you remember that periodic table in chemistry that had like 300 gazillion different chemicals on there, that was, you know, these numbers were the atomic number and the atomic mass. Okay, for the atomic mass or the atomic weight, they're both the same. It's indicated by really the mass of the nucleus. So again, you'll add on the neutrons plus the protons. But again, usually the neutrons is equal to the number of protons. Okay, but sometimes these number of neutrons are variable, okay? And that would give us different isotopes of the same atom. So I'll give you an example, okay, for carbon atom, okay? C12, that's the stable kind, meaning it has 6 protons and 6 neutrons. So your atomic number is 6, and the atomic mass, you'd have to add them together, would be 12. But we also have a carbon-14. Well, the number of protons can never change, okay? And that's why it's always a carbon, because if it has 6 protons, then it's a carbon. So number of protons are the same. But that variation from 12 to 14 is due to the change in the number of neutrons. So now we have 8 neutrons. And those extra 2 neutrons are going to make the C14 an isotope. It is a radioactive isotope due to the variation in the number of neutrons. Okay, so again, your number of protons cannot ever change, okay, for each atom. The number of electrons can, the number of neutrons can as well. And if we change the number of neutrons, that can give us different isotopes. And some of these isotopes are radioactive, and we can use them in science to detect or to diagnose different diseases and brain function and so on. Now, what is an ion? An ion is an atom that either gained or lost an electron. So an ion would have a charge to it. If it gained electrons, which are negatively charged, then we will be making these negatively charged ions known as anions. If it lost electrons, okay, these ions will become positively charged ions. We'll call those cations. And in order to remember which one is positive, which one is negative, cats are kind of like positive, they give you these positive vibes. So those are your positive ions. Anion, an means no. Okay, so this is a negatively charged ion. Now, things that will give you ions can dissolve in water. Okay, and we'll understand this a little bit more when we talk about the water molecule. When they do dissolve in water, for example, sodium chloride, which is table salt, when sodium chloride dissolves in water, it'll dissolve into a positive sodium in a negative chloride. Now that solution has negative charges and positive charges, and it will allow for the conduction of electricity, and hence the name electrolytes. Okay, that's where that term comes from, that when you dissolve something in water and it can conduct electricity, meaning now we have negative and positive charges, that we refer to that as an electrolyte. So you'll find that a lot of salts, we kind of interchangeably use the terms salt and electrolytes because of this. When they are not dissolved in water, so sodium chloride together, we'll call it a salt. When it's dissolved in water, we can now call it an electrolyte. These are different ions that are found in the body. You don't really have to worry about, knowing all of them. We'll kind of know them as we go. So for example, hydrogen ion, you see here that it's positively charged. Chloride ions are negatively charged, and so on. Again, it's not a chemistry class, so you don't really have to worry about it. But what you do want to know is, like, how is this negatively charged? Okay, because it gained an extra electron. What about this calcium? How is it positively charged? It has it's two positively charged because it lost 2 electrons. Okay, so you want to understand what these different symbols mean. Now for a molecule. So we've been talking about the atoms. Now if we take 2 or more atoms and combine them together, that will make a molecule. Okay, we usually do these different formulas so that we could understand what each molecule is made out of. So for example, C6H12O6, which is a glucose molecule, it is made out of 6 atoms of carbon, 12 atoms of hydrogen, and 6 atoms of oxygen. Okay, so the molecular formula again gives you an idea of what the molecule is made out of. Just a quick thing about molecules, that molecules is kind of like this bigger umbrella that has an A and B underneath it. So elements, for instance, would be two atoms combining together, but they are the same atom. So one hydrogen plus another hydrogen atom combining to make a hydrogen molecule. That is known as an element. But if the atoms are different, so for example, we have 3 different kinds of atoms combined together to make this bigger glucose molecule. This molecule is also known as a compound. So again, if the atoms are the same, it'll make an element molecule. If the atoms are different, it'll give you a compound molecule. Now for the different chemical bonds. So how do these atoms connect to each other? And a lot of this occurs due to electrons. So we'll see that we could either transfer electrons, ions, the cations and anions. We've seen how they either gain or lose electrons, or there could be sharing of electrons. And these give you different kinds of chemical bonds. So the first one we'll talk about would be the ionic bond. Then we'll go on to talk about nonpolar versus polar covalent bond. And then the last to talk about are hydrogen bonds. Okay, covalent bonds are one of the strongest bonds in the body, while hydrogen bonds tend to be the weakest bonds in the body, although they're very important, and we'll see their importance when we get there. So an ionic bond is basically the bonding between a negative ion and a positive ion. So just like sodium chloride, sodium was positive, chloride is negative, and because positive and negative attract, they will give you that ionic bond. Okay, again, these ionic bonds do dissociate in water. So sodium chloride or salt does dissolve in water, and again, it can conduct electricity, and hence again we call it an electrolyte. Okay, so anything that has an ionic bond has the ability to dissociate in water. Now the covalent bonds are a little bit different. We're still going to bind together because of electrons, but in covalent bonds we are sharing electrons. And as I mentioned before, covalent bonds are the strongest chemical bonds that we have, and these are due to sharing of electrons. So in ionic bonds, there was a loss or gain of electrons. In covalent, they're shared. So you can see here, this is one hydrogen atom, and this is another, and they are sharing their electrons together. This is the same hydrogen molecule as well. And because they are equally sharing the electrons, and then we call this a nonpolar covalent bond. So nonpolar means, let's understand what polar is, and then we'll, negate it. So polar means kind of like a battery where there's a positive end and a negative end. In a nonpolar molecule, there are no positive and negative ends. Okay, so this hydrogen molecule is neutral, and everywhere you go in this molecule, it's all neutral throughout the whole surface. Same with an oxygen molecule. Okay, you can see here that they're sharing a total of 4 electrons, so 2 from each oxygen atom, and they are equally sharing these electrons. Hence again, there is no positive end or negative end, and with these, this is known as a nonpolar covalent bond. Now if you look at this water molecule over here, it kind of looks like a Mickey Mouse thing, made out of H2O, so 2 hydrogen atoms and a bigger oxygen atom. Now the hydrogen atoms are smaller compared to this huge oxygen atom. So they are going to share electrons to make that bond, but it is unequally shared. The oxygen atom, being the strongest, will pull those shared electrons towards it a little bit. So these shared electrons tend to orbit around the oxygen nucleus a little bit longer than it would orbit around the hydrogen atoms. And therefore, because these shared electrons are pulled towards the oxygen side of water molecule tends to be negatively charged, very slightly so, while the hydrogen ends tend to be very slightly positively charged. Okay, now that is why we call it a polar covalent bond. While the whole water molecule is neutral, but the hydrogen ends are slightly positive, while the oxygen ends are slightly negative. And that ability of oxygen to pull those shared electrons towards itself is known as electronegativity, which I believe is on not the next slide. Okay, so it's on there somewhere in the slides. So the nonpolar covalent bonds, again, whenever you mention covalent, you want to kind of have that reflex where covalent means sharing of electrons. If the electrons are shared equally, that means it's nonpolar. I do not have a positive and negative end of this molecule. You can see here again this chloride. These 2 chloride ions, they are equally sharing these the electrons in the middle. Hence, it is a nonpolar covalent bond. And that is because the electronegativity, or the power to pull those electrons towards themselves, is equal. Okay, they are equally strong. Therefore, the sharing is going to be equal. Okay, it's kind of like hand wrestling, where if the 2 wrestlers have the exact same strength, and they're kind of locked in that position where neither one of them is winning. Okay, so these electrons again are shared equally. They're locked where the strength of these 2 atoms are exactly the same, and neither one of them is able to pull the electrons towards itself. Okay, again, we call this that they have similar electronegativities. Okay, these nonpolar molecules tend to be less soluble in water. Okay, in order to dissolve in water, and we'll learn about this more throughout the course, is that in order to dissolve in water, you want to either be an ionic bond like sodium chloride, or a polar molecule, meaning that you have a positive and a negative end. So these nonpolar molecules are very hard to dissolve in water. And we'll find that this is extremely important when we start learning about how different cells communicate with each other, how do these hormones and chemical messengers reach different cells. And so this is really something you want to keep in mind, that in order to dissolve in water, a molecule would want to either be ionic, okay, or polar. So a nonpolar molecule is very hard to dissolve in water. But they are important, okay, meaning that these can form barriers between cells. So these nonpolar molecules will find these lipid bilayers that are found within the cell membranes and the intracellular organelles. So the walls of mitochondria, the walls of Golgi apparatus, and so on are going to be made out of these nonpolar molecules. So it's a good thing that they can't dissolve, because if everything could dissolve in water, then how can we be kind of solid if we're just going to dissolve within our own water? So these nonpolar molecules, again, are good barriers between different things, and so they'll be used in making the lipid bilayers of the membranes, of cell membranes, and the intracellular organelles. And because, again, they cannot dissolve in water, they'll help in being carriers to other things. Now for the polar covalent bond, again, you have bigger molecules with more electronegativity to them. These are going to pull the electrons towards themselves, just like we were talking about that water molecule where the oxygen is huge. The electronegativity of oxygen is much higher than that of hydrogen. And so they're still sharing electrons, but their sharing is not equal. The oxygen takes the electrons a little bit more and longer than the hydrogen do. So the oxygen side of water is negatively charged, while the hydrogen side of water is positively charged. And we call that a polar covalent bond. And you can see here in this picture, so water is really the infamous example of polar covalent bonds, where you have on the oxygen end a very slight negatively charged side, while on the hydrogen ends you have a very slight positively charged. Now, and this is why in order for a substance to dissolve in water, it has to have polarity to it. So sodium chloride has sodium positive and a negative chloride, and that's what makes it easy for it to dissolve in water, okay, because of the polarity of the water molecule. So you see that polar molecules can dissolve in blood, can dissolve in interstitial fluid, which is basically the fluid between tissues, and can also dissolve in intracellular fluid, so the fluid that's inside of the cells. So you'll see here that covalent bonds are really important. Nonpolar are important to kind of make that barriers, like the cell membrane wall and the walls of the organelles, while these polar covalent bonds are important so that things can dissolve, like we can dissolve salt, we can dissolve glucose, we can dissolve different things in there and be able to distribute them throughout the body. Now for hydrogen bonds, and these are the weakest bonds within the body, and it really ties in, I want to see if I have it, there we go. So it really ties into the polar covalent bond, okay? I'm going to go to the next slide to kind of explain what I just said. So these hydrogen bonds are related to your polar covalent. So this is one water molecule, and here's another water molecule. Remember we said that the oxygen side is very weakly negatively charged, while the hydrogen end has a very weak positive charge. Now that positive and negative are going to attract, and that right here, that attraction is the hydrogen bond. They're very weak, and you could see that that weakness is kind of, we represent that by these dotted lines. So whenever, as soon as you boil water, these hydrogen bonds break and the hydrogen and your water molecules evaporate. So they're very weak, but they're extremely important because those are the bonds that bind the two strands of DNA together. So you'll find these hydrogen bonds within our genes. And that again, if you want to tie this to clinical, if somebody has a very high temperature, so hyperthermia, the danger of having these high temperatures would be because of the danger of breaking these hydrogen bonds. Again, they're very weak. They can be, high temperatures can break these bonds. And if you break your DNA bonds, that is really not a good thing. So you won't, these bonds cannot be, they're not reversible. Okay, so if these bonds are broken, even if the temperature goes down, there is no way for these bonds to go back the way they were. Now, what are free radicals? So we always hear about, free radicals that cause damage. They cause skin wrinkling, which is true. Okay, so these free radicals, they do lead to a lot of the aging process, like eye diseases like cataract, for example, which is the lens becoming opaque. Cardiovascular diseases, so, coronary artery disease and so on. Neural diseases, most of the aging process is really related to the presence of these free radicals and the body not being able to deal with them. So what are free radicals? Free radicals are atoms or molecules that have an unpaired electron in the outer orbit, surrounding the nucleus. So remember we said that in order for an atom to be stable, you want that outer shell to have the maximal number of electrons allowed. So if the outer shell needs to be filled up with 8 electrons, then if it has 7 or 6 or 5, anything less than 8, it will not be a stable electron. And it will start shedding off these extra electrons, so removing those electrons, and that will make it a free radical. So it's that, these loose electrons are what are really dangerous. Okay, so a free radical would be an unstable atom that is shedding electrons everywhere. It is important though, this is how white blood cells destroy pathogens. Okay, so we can use it to our advantage, but if it is, if we're exposed to things like radiation or toxins, okay, the accumulation of a lot of free radicals that would building up in the body where our bodies are unable to deal with that accumulation, that can start leading to these different aging processes that we see. So if you want to stop the aging process, you want to be able to deal with these free radicals. And that is something I tell all my students, if you ever figure out something, say 15, 20, 30 years down the road, if I'm still here, let me know. Okay. Examples of biologically important free radicals are superoxide anions. So this O2, hydroxyl radical, so OH, and nitric oxide or NO. And these dots, okay, those indicate that this is a free radical. Okay, so that would lead to the destruction of the surrounding cells. Now at this point, this would be a really good time to pause, relax, and come back. Okay, so welcome back from break, and we will move on. Okay, so we'll talk a little bit about solutions, acids and bases, and so on. So solution, which is basically anything that if you can dissolve something in water or any other liquid, that will give you a solution. Okay, so what you are dissolving is known as a solute, and the liquid is known as a solvent. Okay, so if you're dissolving salt in water, the salt is your solute while the water is the solvent. Okay, and since water makes about 60% of our total body weight, again, in order to be transmitted through the water stream or through our bloodstream, you want things to be able to dissolve in water. And a lot of the chemical reactions that happen within the body, so a lot of the metabolic reactions, they do involve water, whether the removal or the addition of water molecules. So we'll see reactions that involve water, and either we are removing a water molecule, and that is known as dehydration synthesis. So you are making something by the removal of water. So that's, the literal translation of dehydration synthesis. So for instance, you have these two building blocks. So monomer means a building block. Mono means one. So this is one building block and this is a second one. And in order to tie them together, you will remove a water molecule. Okay, and now you have that covalent bond between these two monomers. And, you did that by the removal of this water molecule, hence the name dehydration synthesis. In order to break this bond, okay, all you have to do is add that water molecule again. So this is a reversible reaction. And the addition of water is known as hydrolysis. So you can take this bigger molecule and break it down into its monomers by the addition of a water molecule. And again, hydrolysis literally means hydro means water and lysis means to break. So you broke that covalent bond by adding a water molecule. And you'll see that a lot of the, dehydration synthesis and hydrolysis, these are things that the body uses in order to either digest or to build up things like different proteins and so on. So the solubility in water, remember we said some things or some molecules are soluble in water. And because they like water, they are known as hydrophilic. So hydro again is water, philic means the tendency to like something. So these hydrophilic molecules are either polar molecules or ionized molecules that can dissolve in water. So remember we said like sodium chloride, because it has that positive sodium and negative chloride, it is able to dissolve in water. So again, in order to dissolve in water, you want the molecule to either be ionic or polar. Okay, these are extremely important points to know because again, it's going to help us so much when we talk down the road in this course, when we talk about chemical signals and so on. These points are extremely important to keep in mind. While the molecules that are made out of non-polar covalent bonds, so there is no positive and negative end to the molecule, these are known as hydrophobic. So you cannot dissolve these molecules, or it's very hard to dissolve them in water, which, we kind of translate into water-fearing molecules or hydrophobic molecules. So we'll take an example, for instance, triglyceride, which are fat molecules. These molecules are hydrophobic, okay? So fat molecules will not be able to dissolve in water. Now, what are amphipathic molecules? Amphipathic comes from the term amphibian. So you have the amphibian reptiles, for example, that are, like to go in water and can also go on land. So these amphipathic molecules are the same thing. These are bigger molecules that have one side of the molecule is polar, which is hydrophilic, and the other side of the molecule is nonpolar, meaning that it's hydrophobic. So please tell me there's a picture here. Okay. And I'll go back to that slide in a minute. So you can see here, this big molecule, one side of it has a polar head, while the other part, which I'll just call the tail, is nonpolar. Okay, that makes this molecule amphipathic. While your water molecule, if you remember, is polar. Okay, so this, in order for an amphipathic molecule to go within the bloodstream, to be transported in blood, you would have to get these polar heads in contact with the water molecules and hide all of these hydrophobic tails in the middle. So this is how an amphipathic molecule can be transmitted in water. It makes these little clusters where the polar heads face the bloodstream and these nonpolar hydrophobic tails cluster within the middle, okay, hiding away from water. So you'll find that, some of the, for example, phospholipids, which is a kind of fat, that is very important in making cell membranes. This is how they are transported throughout the bloodstream. Okay, again, these spherical clusters where your polar heads face the water and these non-polar heads, or the hydrophobic tails, are hiding in the middle of the cluster. Okay, and that arrangement, as I said, is really important, especially when you're making cell membranes. Okay, so you'll see that it's important in the formation of your plasma membrane and how we absorb some of these nonpolar molecules and transport them in the blood. So absorption through the intestinal wall and transmitting it through the bloodstream. Sometimes we're going to make these spherical clusters. Now, the difference between an acid and base is that an acid has the ability to release hydrogen ions, while a base or an alkaline has the ability to accept a hydrogen ion. The more hydrogen ions released by the acid, the stronger that acid is. Compare that to a base. The more hydrogen ions that can be accepted by the base, the stronger that base is. The hydrogen ion, we don't really talk about hydrogen ion concentration as a number, but it is expressed in the logarithm of that number, and we call that the pH. The range of pH in humans is 7.35 to 7.45. So the pH scale out of humans, okay, is from 0 to 14, where neutral is right in the middle, which is distilled water, right in the middle at 7. Any number below 7 would be acidic. Any number between 7 and 14, so higher than 7, would be alkalinic. The normal range of pH in humans is 7.35 to 7.45. So our bloodstream tends to be a little bit on the alkalinic side. Know that though, in different parts of the body, the pH is going to change. So the pH, for example, in the stomach can reach 3. In some reports are like, oh, the pH is at 1 because of the hydrochloric acid that is being made. But the overall, the pH of blood is this range, and this is a range that you want to be familiar with. Now the terminology of different things. So we kind of talked about chemistry and biochemistry and so on. We always hear the term organic versus inorganic. So organic chemistry is the chemistry, studying molecules that have tons of carbon in it. So for example, we'll see like steroids, cholesterol, glucose, those have lots of carbon, okay? And that would be dealing with organic chemistry. Inorganic chemistry would be the study of molecules that do not have carbon. Okay, but, for example, CO2, which is carbon dioxide, it has one carbon molecule or one carbon atom in there. It's still considered inorganic chemistry because you want to have lots of carbon in order to be considered organic. Biochemistry is really the chemistry that happens within our biological cells, okay? That's where the term comes from. So it's the chemistry of living organisms. And it is really a branch of organic chemistry. So we want to talk about the organic molecules that we have in the body or we deal with somehow. So there are carbs, and under carbs we'll talk about polysaccharides, disaccharides, and monosaccharides. For each of these, you want to know, the polymer, which is the bigger molecule, and what are they made out of? So what are their monomers or their building blocks? Okay, and obviously the function of each of them. So carbs, for instance, polysaccharides are your bigger molecules. Poly means a lot. Saccharide means sugar. So these polysaccharides, we can eat polysaccharides like starch, but we cannot absorb it. We have to break it down into its building blocks. So we break it, the digestive system will break it down into disaccharides, which are made out of 2 sugar molecules. We still cannot absorb that. The small intestine only has the ability to absorb monosaccharides, which are, made out of 1 sugar molecule. Excuse me, my allergies going crazy here. Okay, so we were saying, that the body can only absorb these monosaccharides, and that last stage of breaking down the disaccharide into a monosaccharide in order to absorb it is actually performed within the intestinal cell, in the small intestine. So you see that throughout the whole digestive system, we're going to start breaking down the starch into its disaccharides, and your last, sometimes the very last stage of breaking down the di into a monosaccharide can actually even happen within the intestinal cell. Now for lipids, there are three of them that we want to learn about. There are triglycerides, and triglycerides, we'll see that they're made out of fatty acid chains and glycerol. Triglycerides is just a fancy term for fat. Okay, so the fat cells are made out of triglycerides. Phospholipids are made out of fatty acids, glycerol, and others. I'm kind of going over this kind of fast because we're going to go in detail in the next couple of slides. And then steroids, we'll see that they're technically all kind of spin-offs of cholesterol. And then your protein, which is a polypeptide chain with building blocks of amino acids. And then our nucleic acid, which are DNA and RNA, and those building blocks are known as nucleotides. So let's talk about the carbs. And I want to go to the polysaccharide first. So this is what we eat. Okay, we eat a whole chain. Each one of these little tiny beads is considered a sugar unit or a glucose unit. And you see them here all, attached to each other to make this polysaccharide chain. This is how we either eat it or store it. Okay, so when we store, if we intake too much sugar or too much starch and we want to store this, our body stores it in the form of glycogen. And glycogen is stored in the liver and in our skeletal muscles. Skeletal muscles, if you're not familiar with that term, are the muscles that are attached to the skeleton, hence the name skeletal muscle. So again, glycogen is how we store glucose, and we store that in the liver or in the skeletal muscles. And you'll see here that it's made out of these units of glucose. So this ring right here, this is one glucose unit, and this is another, and they're attached to each other to make that glycogen storage molecule. Okay. Here you can see a disaccharide, okay, made out of 1 and 2 molecules. And this is a sucrose molecule, which is basically table sugar. But in order, again, our body does not have the ability to absorb a disaccharide. Inside of our intestinal cells, we have an enzyme known as sucrase enzyme, ASE. The sugar is sucrose, but the enzyme is sucrase with an A, and that will break it down into glucose and fructose. These are the building blocks. Both of these are monomers, are monosaccharides, and the intestinal cells can absorb them. Okay, and you see here that we break down that bond by the addition of a water molecule. So this was, we break it by hydrolysis. Okay, so this equation is actually showing you the buildup of it, but during digestion, we are going to break it. So again, remember, all of these chemical reactions are reversible. Okay, so now how does the body use sugars? So again, we eat polysaccharides. We have to break them down into di and then monosaccharides, which, by the way, kind of mentioning this sucrose sugar, which is table sugar, but there's also lactose, which is milk sugar. And we also have, within our intestinal cells, we have an enzyme known as lactase enzyme that will break down lactose into its monomers. In some individuals, lactase enzyme is deficient or even totally lacking, and that would lead to lactose intolerance, meaning that if they eat or drink lactose, which is milk and milk products, that has that sugar, they will not be able to digest lactose into its 2 building blocks. And that will lead to accumulation of that kind of sugar. which will ferment, leading to distension, abdominal distension, and gas and flatulence, which is the passage of gas, and can even lead to diarrhea and so on. And that is again because the body does not have the ability to absorb a disaccharide. Okay, so this again is kind of tying what we're learning here into different clinical scenarios. Now, how do our bodies use sugars? So the glycogen is how we store the extra sugar. We'll take those sugar molecules, the glucose monomers, bind them together to make glycogen. And again, remember, we store glycogen in the liver and in our skeletal muscles. So whenever we need, when, say 3, 4, 5 hours of, non-eating, okay, so the body is now kind of in a period of fasting or in the state of fasting. Please note, and we'll talk about this when we talk about the digestive system, that the act of us, or the trend of snacking all day, really has messed up a lot of the digestive system and a lot of how these metabolic reactions occur. So normal quote unquote eating would be to have your meal with no snacking in between. So when we say that during periods of fasting, that would be in between meals. So, nothing from breakfast to lunch, no snacking there. And this is when the glycogen that we have stored within the liver and skeletal muscles will start to break it down into glucose in order to burn it and use it for energy in between meals. Okay, and glucose, we all, kind of by default, that would be your blood sugar, since most cells need glucose to burn in order to produce energy. Now, next on our list are lipids. So lipids are molecules that are made out of mainly hydrogen and carbon with other things. They are linked by nonpolar molecules. So now hopefully in your head you kind of have this reflex. Whenever you hear nonpolar, means that it cannot dissolve in water, which means it's hydrophobic. Okay, the lipids that we have fatty acids. They tend to be in chains. We also have triglycerides, phospholipids, and steroids. They are really important sources of energy. They actually release more energy than glucose or than carbs do. The only thing is that our body has to deplete and get rid of all of the glycogen storage first before it starts breaking down lipids. And that's, the whole concept of kind of dieting, that dieting needs to make sure that somehow you are depleting and using up all of the stored glycogen, and then you start tapping into the fat cells to release energy from fat. Okay, so it's a very valuable source of energy. Lipids are really important for cellular membranes and important signaling molecules, which means how cells communicate together. And we'll see here what a fatty acid chain looks like. So fatty acid, this is one chain which has like all of these carbon atoms with lots of hydrogen. Okay, there could be other stuff, but mainly carbon and hydrogen. And this is a saturated fatty acid. So you'll see here that there's just one covalent bond between all of these carbon atoms. These saturated fatty acids tend to be solid at room temperature and are of animal origin. While unsaturated fatty acids, you'll see that one or more carbon atoms can have double bonds, double of these bonds here, these covalent bonds. Okay, these unsaturated fatty acids tend to be liquid at room temperature and are of plant origin. So for example, olive oil, corn oil, and so on. So which one of these do you think is healthier? Is it the saturated or the unsaturated fatty acid? So it is actually the unsaturated ones that are healthier for our body. Okay, and depending on how many of these double bonds we have, so if this one, for example, has 2, so we'll call this a polyunsaturated fatty acid. Had it had only one of them, we would call it a monounsaturated fatty acid. Now, triglycerides, we could also call them triacylglycerols. That again is a fancier term for fat. Okay, it's made out of a glycerol molecule with 3 fatty acid chains, hence the name triglyceride. So again, a glycerol molecule with 3 fatty acid chains. These are found, you'll find them in the body, in the blood. They're made by the liver. Okay, so we will realize that somebody that has liver failure, for example, will kind of have abnormalities in their formation of triglycerides. Okay, and we store these triglycerides within our adipose tissue. Adipose tissue basically is the subcutaneous fat. We have the ability to store lots of energy. And again, during fasting periods or exercise, for example, once we are done with our glycogen storages, we'll start tapping into the triglycerides. Now, okay, go back to the triglycerides for 1 second. These are totally hydrophobic. Okay, there's no way that these can dissolve in water. They are important structures, but we have no ability to dissolve them in water. Compare that with a phospholipid. So phospholipid is made out of a glycerol portion, 2 fatty acid chains, and in order to make it to make it into an amphipathic molecule, there is the addition of this phosphate portion, so PO3, which is phosphate. Now we have a hydrophilic head, which is this right here, hydrophilic, which is water-soluble head, and these 2 hydrophobic tails that represent these 2 fatty acid chains. Okay. So the phospholipids is an amphipathic molecule because we have one side of it that's hydrophobic and the other side that's hydrophilic. And these are extremely important in the formation of the lipid bilayer of all of our cell membranes. Okay, so these phospholipids are extremely important within the structure of the body. So this is another good time for you guys to stretch, to pause, stretch, get some water, do jumping jacks, whatever you need to do to come back. Okay, so welcome back, folks. Now we'll talk about steroids, and this is kind of like a subtype of lipids. It is the skeleton of it is made out of these interconnective rings of carbon. You'll see 4 of them connected together to make carbon, and usually this is kind of like how cholesterol is made, and then we'll use that cholesterol to make different things. Okay, so we could use it to make sex hormones like estrogen and testosterone, or we could make cortisol, which is secreted by the adrenal cortex, and so on. So again, cholesterol is basically your kind of like baseline for steroids. And using that cholesterol, we could make it into these different things. These are obviously not water soluble. Now proteins, which is, we've got proteins and nucleotides to talk about. So proteins are made out of carbon, hydrogen, and other things like oxygen, nitrogen, or sulfur. And these molecules are macromolecules, so these are big molecules made out of, hundreds and thousands of little tiny atoms. Our body has the ability to synthesize a lot of amino acids, but some of them we have to take in our diet. So we cannot make all of them. The ones that we have to take from food are known as essential amino acids. The ones that we make in the body are known as non-essential amino acids. So what is the structure of a protein? We break it down into 4 different levels. Really, the primary structure of the protein is determined by the sequence of these amino acids. So the building block of a protein is known as an amino acid. And these little tiny beads, each one of these represents one amino acid. And you'll see here that they attach to each other to make a chain, what known as a polypeptide chain. And the bonds between these polypeptide these amino acids are known as peptide bonds. And so your primary structure is basically the sequence of the amino acids. And we have that manual on how to make these proteins in our DNA. Okay, so that is your primary structure. But that primary structure, which is again basically, a long train of amino acids, that is not how proteins are found in our bodies. That string of beads has to change its shape so that it can perform different functions and it can be transported in the bloodstream. And that final shape of the protein is known as a conformation. So the protein has to change its shape, and that final shape is known as conformation. So now we already know what the primary structure of a protein is. Now, what is the secondary structure? Well, secondary structure would be that some of these, let me see, where's the better picture? There we go. So your primary structure, again, is basically the sequence of amino acids. And if anything changes in that sequence, that can lead to a mutation and can lead to different diseases. And we'll talk about that, in a second. The secondary structure would be the formation of hydrogen bonds between different amino acids. And that could lead to either an alpha helix, so basically twisting, or what is known as a beta pleated sheet. It's kind of like when you make a fan out of a sheet of paper. So these hydrogen bonds tend to happen in between amino acids that are not neighbors. So we call them non-adjacent amino acids. Okay, so again, these hydrogen bonds between non-adjacent amino acids can lead to the secondary structure of either an alpha helix or a beta pleated sheet. Now that we have either one of these sheets, the alpha helix or the beta pleated, we can start making a tertiary structure, which are basically more bonds between different amino acids. And it's a very, very 3D shape known as your tertiary structure. Most proteins tend to function and stop the process at a tertiary structure. Some proteins, though, are huge macromolecules and would have to go through what are known as a quaternary protein structure, or level 4. And we call that a multimeric or many parts protein. So you can see here that this is a hemoglobin molecule, and it is made out of 1, 2, 3, 4 molecules of protein put together. Okay, so we'll take 4 of these tertiary structures and attach them together to make this huge hemoglobin molecule. Okay, that would be level 4. Remember again that most proteins tend to end at 3. Some do, though, have to go to that 4th level. Now, hemoglobin, as I said, is an example of that multimeric protein that goes all the way to level 4. It's made out of 4 subunits. And you can imagine that a mutation or a change where one amino acid has been switched at the level of the primary level, so in the making of that primary polypeptide chain, where a molecule of valine is an amino acid, replaces a molecule of glutamic acid, which is another amino acid. Okay, that can lead to a disease known as sickle cell disease. So sickle cell disease is a disease of the red blood cells where there's just a mutation, just one simple replacement of one amino acid instead of another. And when these individuals are exposed to low oxygen levels, their hemoglobin changes from a disc shape to a sickle shape. And that could lead to blocking of the small capillaries. And it in some individuals, if it's severe, it's an extremely painful kind of disease. So we'll see here, this is what your normal red blood cell looks like, and this is what a sickled, this is what a sickled RBC looks like. So here are normal ones, and here are sickled ones. These normal ones are very malleable. You can see them here kind of going within the blood vessels and can squeeze themselves within the capillaries. That ability is lost if the red blood cells are sickled, and they tend to block capillaries and lead to hypoxia or low oxygen levels in those areas. And again, it can be extremely painful. And I do have this little video that you guys can watch, to get more information on sickle cell. Now, last but not least are nucleic acids. And nucleic acids are basically DNA and RNA. DNA has the manual on how to make proteins, and that would be found inside of our nucleus. RNA is a structure that helps in protein synthesis. So DNA and RNA work together in order to make proteins. Okay, their building blocks are known as nucleotides, and they are basically, each nucleotide is made out of a phosphate group, a sugar, and a base. So we'll see here, this is a DNA molecule. So DNA stands for deoxyribonucleic acid. And the sugar is deoxyribose. You have phosphate, and then you have a base, which we'll talk about the differences in bases in a bit. In RNA, the sugar is a ribose sugar. Phosphate is the same. And the bases we'll talk about as well in a bit. And they have some similar bases, but they differ in one. You can see here DNA, again, forms our genes. And DNA is usually double-stranded, so there are two different strands, and they connect together through these little bases here through hydrogen bonds. Now, these bases can either be purines, and the purines we have are either adenine and guanine, or they can be pyrimidine or pyrimidine bases, which are cytosine and thymine. In order for them to attach like pieces of puzzles, the hydrogen bonds have to happen between a purine and a pyrimidine. And the rule of thumb is that adenine will form 2 hydrogen bonds with thymine, while cytosine will form 3 hydrogen bonds with guanine. And that is, will go throughout the whole DNA strand. So you'll see that C and G, which stand for cytosine and guanine, and then T and A, standing for thymine and adenine. And you'll see that throughout the whole strand. So DNA, again, is double-stranded. The sugar is deoxyribose, and your bases are these 4 bases that we just talked about. RNA, on the other hand, is a single chain of nucleotides. Okay, so it's not double-chained like DNA. It's just one single chain of nucleotides. The sugar is ribose sugar. And for the bases, they're all the same except RNA does not have thymine. RNA has uracil instead. Okay, and so if you read a sequence of bases and uracil comes up on there, you would automatically know that this is a strand of RNA. While if you're looking at a sequence and a base, you see a T or a thymine base, well then you automatically know that this is a strand of DNA. Okay, the 3 other bases, adenine, guanine, and cytosine, those are found in both DNA and RNA. Okay, so that was the end of this chapter. Let me know if you have any questions.